My name is Alyssa Cavezza, and this is the assay for proteolytic activity using azocasein. Just for some background, an enzyme is a type of protein that catalyzes reactions. Enzymes lower the activation energy of the reaction in order to speed it up. In living organisms, this plays a major role of speeding up the reactions that happen in daily life, such as metabolism reactions. It also allows for the regulation of different reaction rates in the body. The enzyme that was used in this experiment was trypsin. Trypsin is an enzyme that is found in bacteria and the small intestines of many animals. This enzyme helps with digestion of proteins that were eaten by the animal. The reaction catalyzed by this particular enzyme is protein hydrolysis. The enzyme cleaves after the proteins ARG and 2YR. The difference between bacterial trypsin and animal trypsin is that bacterial trypsin is secreted, while animal trypsin is in the intestines. For our question for this specific experiment was, at what pH does the enzyme trypsin have the highest activity? Our hypothesis for this question was when the bacterial enzyme trypsin is placed into a basic 8 to 9 pH, it will have the highest enzyme activity. The rationale for this hypothesis was that because trypsin originates in human/living animals' small intestines, which tend to have a basic pH of 8 to 9, enzyme activity will likely be highest at these pH levels for the experiment. Our experimental variable for this experiment was pH. We had 3 replicates for each pH ranging from 5 to 10, so we did 5, 6, 7, 8, 9, and 10. The control variables were that the solution was at a salt concentration of 0.15 molar and the temperature was 37 degrees Celsius. For our negative control, we left out trypsin and added an additional 50 microliters of the reaction buffer to the solution to maintain the volume. We had 1 negative control for each pH 5 to 10. For the procedure of this experiment, in the beginning, in our centrifuge tubes, we added 450 microliters of the 0.15 molar NaCl reaction buffer, 500 microliters of the substrate azocasein, and another 500 microliters of the enzyme trypsin. For our negative control, we left the trypsin out. After adding the enzyme, we incubated the tubes in a water bath of 37 degrees Celsius for around 20 minutes. After we added 50 microliters of TCA, stopped the reaction from continuing, and precipitating the undigested substrate, we then centrifuged the tubes at 12,000 RPM for 2 minutes, and again at 14,000 RPM for another 2 minutes, to separate the undigested substrate from the supernatant and basically form a little pellet at the bottom of the centrifuge tube. Then we transferred the supernatant from centrifuge tube to cuvettes and observed the absorbance of each tube by setting the spectrophotometer at 440 nanometers. The substrate we used in this experiment was azocasein, which is a milk protein. The random addition of sulfonylulamide within the substrate has a positive correlation to the absorbance measurement at 440 nanometers. Our results were that from pHs 5 to 8, the averages increased, while at 9 and 10, it then decreased. Our standard deviations were less than 0.01, indicating that we had a very low variability. Our t-tests were also less than 0.01, indicating that all of our solutions were statistically significant. As you can see in the bar graph, from pHs 5 to 8, the pH is increasing, and it reaches the highest peak at 8, and then decreases from 9 to 10. 3 replicas were performed for each pH in our experiment, and our p-value was less than 0.05 for the t-tests. Our observance values in this experiment was that as the pH increased, the color of the solution went from a lighter color to a darker color, orange red with the enzyme, and then a clear yellowish after we centrifuged it. This experiment, we concluded, was adequate to test the hypothesis. The enzyme activity was measured in absorbance by the spectrophotometer. Our conclusion was that yes, the hypothesis was supported by the results. From the pH of 5 to the pH of 8, the absorbance increased and was at its highest peak at pH 8, at the absorbance of 440 nanometers. The absorbance then decreased as the pH went from 9 to 10, proving that the enzyme activity was highest at the neutral pH of 8 as our hypothesis stated. If we were to add any changes to this experiment, and we were to perform it again, we would perform 3 negative controls instead of 1 each pH instead of just 1. It is debatable whether or not our results got us the best enzyme activity, as the other groups that did pH got pH 9 instead of 8 for the optimal. Our hypothesis stated that pHs 8 to 9 were optimal, so their data does support our hypothesis. However, another group got the results of ranging from 10 to 11, indicating that they believed that a higher pH would give the best results, and this did not support our group's results. And this is the assay for the proteolytic activity using azocasein.